Assalamualaikum and hi everyone. So we want to present to you about application of micropropagation in Malaysian crop production. So this is our group members. First, my name is Ainin Sofia binti Rosli and my members is Wasilah Husna binti Rahmat and Nur Harmiza Izzati. For our problem statement, application of micropropagation, also known as plant cell culture on Malaysian crops, has become an important strategy to increase food security as well as economy. Identify a few important crops that utilize such plant biotechnology. Describe the basic concept behind the technology and explain the various types of the micropropagation. Describe the strengths and weaknesses of the application. As for our objective, we have 4 objectives, which is first, to identify a few important crops that utilize such plant biotechnology. Second, to describe the basic concept behind micropropagation technology. The third one is to explain the various types of micropropagation, and the last one is to describe the strengths and weaknesses of this application. As for our introduction, first is micropropagation of plants is a useful technique for large-scale propagation of crops, medicinal and ornamental plants, tree species and other economically important plants. As for short, micropropagation is the artificial process of producing plants vegetatively through tissue culture or cell culture techniques. In the artificial process of propagation, plants are produced in vitro by asexual means of reproduction by vegetative propagation. Micropropagation are the valuable method for rapid production of disease-free plants, rapid multiplication of rare species, genetic transformation of plants, and production of plant-derived bioactive compounds. Plants can be generated sexually by the process of seed production or asexually through methods of the multiplication of vegetative elements. For the clonal propagation or the process of producing genetic copies of plants is one of the methods of asexual reproduction that allows plants to be multiplied from a single individual seed. The only means for in vivo propagation of certain plants is asexual propagation by propagation of vegetative parts because they do not produce functional seeds as observed in figs, grapes, bananas and many more. So, successful application of clonal propagation has been observed on potato, apple and many other ornamental plants. So this is the example of Agrotech Center for plants development in Malaysia. First, we have Sarawak Biodiversity Center. Second, we have FAMA, which is Federal Agricultural Marketing Authority. The last one is Kulim, which is Kulim Agrotech Center in Kulim, Kedah.
Let's hit off with important crops utilizing micropropagation. First is oil palm known as Elias guineensis, which was essential for Malaysia's palm oil industry. Clonal propagation was made in order to improve its yield. Second, orchids of Phalaenopsis. It is a valuable ornamental plant for exports. It undergoes rapid multiplication to produce desirable traits. Third is banana, which is a major staple crop that needs micropropagation as an efficient production for disease-free banana planters. So, micropropagation involves the aseptic culture of plant cells, tissues or organs in a controlled environment, leading to the production of multiple genetically identical plants. First is initiation. It is an explant that is taken from the mother plant. The surface sterilization was made to eliminate contaminants and it's placed in a nutrient-rich medium for initiation. Second is proliferation, which is actively dividing cells from a callus. The subculturing is made to maintain cell division. Third is rooting, an induction of roots on microshoots to transfer into a rooting medium. 4 is acclimatization, which was a gradual transition to soil conditions and to transfer it to the field for further growth. For details, this is the reference for micropropagation process.
So now I'm going to continue the types of micropropagation. Some of the types that we obtain is meristem culture. Meristem culture works by multiplication to growth and proliferation of existing meristem. It can be through apical shoot exist from the parent plant by multiplication of existing meristem within axillary shoot, which proliferate on explant after removal from the parent plant. The second one is color structure. The formation of individual organs such as shoot and roots either directly on the explant where a preformed meristem is absent or the origin from callus and cell culture generated from the explant is termed as organogenesis. The third one is embryo culture. Embryo culture have a formation of a bipolar structure including both short and root meristem either of the adventative origin or directly from the explant or origin from callus and cell structure, in this from the explant which termed as somatic embryogenesis. In this culture, explant is cultivated in suitable embryo inducing medium and the embryo is cultivated into rooting and shooting medium to obtain a whole plant. Next, I'm going to explain about the step of micropropagation in each type that I just explained just now. So, from this picture, you can see that these three cultures have different process, which meristem culture have multiplication through the growth of the plant, and then waiting for the developed shoot, and finally the plant can be transferred. The example of this process can be dahlia and carnation. Meanwhile, for color structure, a small piece of tissue from the plant and is grown in a natural media under antiseptic condition. Then the tissue will proliferate into callus, and then when a small portion of the callus tissue are transferred to another medium with specific hormone, the differentiation of plantlets is induced. The plantlets then are transferred into a pot or soil and raised to maturity. The example for this process is orchid. And the last one is embryo culture, where it takes a cell from the plant to generate a miniature form of the adult individual after its germination. The example plant that go through this culture is sunflower. Now is the strain of micropropagation. First one is disease-free plant. So when you do micropropagation, it can contain a very low concentration. And then because it has been demonstrated that the shoot apertures of virus infected are frequently devoid of viral particles. The next one is germplasm conservation. So, most seed and vegetative organs have a limited storage life. Research on germplasm preservation has concentrated on the development procedure to extend usable lifespan. So, it also high in cytoplasmic and non-vacuolated, and also meristem is genetically stable and can be generated into pathogen-free plants. The next one is production of artificial seed. Produce an analog to true seed which is based on the similarity with zygonic embryo with respect to growth morphology, physiology, and biochemistry. So there are two types of synthetic seed that has been developed, namely is hydrated and desiccated. So now we're moving to the weakness of micropropagation. The first one is vitrification, can also known as hyperhydration. Such should inhibit a decline in the rate growth of multiplication. So due to the morphological, physiological, and metabolic dearrangement occurring during intensive root multiplication in culture. The next one is cost. So to do micropropagation process, you need a suitable sophisticated facility with trained manpower. Because of it, it can be super expensive. The last one is contamination. Though precautions of high order are normally taken during culture, but there's still a chance of contamination by various pathogens. Contamination could cause a very high loss in a short time.
As per the conclusion of this application of micropropagation, to conclude, this propagation technique need to be publicized to public so that more people will use propagation techniques for crops. Micropropagation has their own benefits to do the propagation. However, propagation also has its weaknesses for doing it. So this lack of success can improve day by day to show that propagation has no weaknesses in the future.
So this is our reference. And thank you for watching. Have a great day.